I am a researcher or clinician interested in a variant and want to know if it's been published before and to understand about that publication landscape. This is by far the most frequent use case for our Mastermind users. There are many others that I described earlier and some more that I'll describe that are a little bit different than that, but this is the most frequent use case where previous to coming across Mastermind, users would either be searching through PubMed or Google Scholar. Mastermind is an excellent replacement of those processes in your clinical or research workflows that gives you vastly superior results in terms of sensitivity and specificity. And so the specific example that I want to walk through is a variant in the UROS gene. It is a missense variant. I'll type in this variant. As I said, if this is the variant that you've encountered in your patient's VCF file or otherwise in your research cohort, or if it's a variant that you have questions about or interested in for other reasons, typing in the gene and then the variant and performing your search will take you to this result page where you can get a landscape view of the diseases that it's associated with. And if you weren't familiar before, it should be apparent now that the UROS gene and this particular variant is associated frequently with porphyrias. We don't need to go into the nature of that disease, but you can see a number of aspects of porphyria and the different types of porphyria that are involved in this particular circumstance. If you want to maximize the sensitivity, you execute the all diseases search. If you want to enhance or hone the specificity of your search, you'd click on any one of these specific disease entities below. In this example use case, it's most typically the case that our users will engage the all diseases search, even as it does contain about 70 total references where this particular variant is described in the medical literature. I'll show you ways that you can hone the specificity of your search so that you don't feel the need to look through all 70 of those references to come to conclusions that you need to for your clinical workflow. So we talked about on the left the variant landscape for a gene-specific search. In this case, this is now showcasing those variants in the diagram that co-occur in the context of the mention of the particular variant that you've searched on. And so what you're seeing is all of the variants that also appear in the context of this particular variant and those references that mention the C73R mutation. Below is the comprehensive list of all of the variants in the UROS gene, just so that you never lose sight of the comprehensive landscape of all of the variants that Mastermind has identified. And we'll talk through a couple of those features here in a moment. But to turn your attention now to the new feature that we haven't talked about yet, on the right side of the Mastermind interface, where on the left side is the variant landscape, on the right side is the publication landscape. And we've depicted it visually here and in list form, just as we have done for the variant landscape. In the upper right is the literature landscape by publication date on the x-axis, and by impact score for the journal on the y-axis. Each of these individual icons represents a specific reference. When I hover over, you can see the title of that reference and in parentheses the journal. The size of the icon is a reflection of the specificity of that content to your particular search. In this case, we've not yet engaged any category terms. We've searched on just the gene and the variant. As you can see in these filter icons, we're casting a wide net for all diseases. We've searched on a gene and a particular variant. As I'll show you in a moment, when we add new category filters, you'll see these icon sizes and therefore the relevance of the content to your search change in accordance with what you've told Mastermind you're interested in seeing. What you can see below is all of that content, all of those 70-some articles that talk about the variant of interest that are visually depicted here. You can zoom in and reset that graph as you please. These are all listed below in the article list pane. If you're interested in collapsing the publication history that's depicted visually, you can collapse that and see a bigger sweep of the articles in list form below. These PDF icons indicate that Mastermind has secured and indexed the full text content for each of those references. So without going into any detail, you can see that Mastermind has a very broad reach into the medical literature. The journal for each of these individual references is depicted, as is the publication date. The title is depicted here, and so too are the number of mentions of the gene of interest and the variant of interest, which is a useful way for you to triage the relevance of the paper depending on your specific type of inquiry. At the top here is the ability to export this PMID list as a comma-separated variant file. That will download for you all of the bibliographic information and preserve the sort order of these references for those of you who want to get the information and do some downstream processing with it and don't want to transcribe this manually. There's the export capability. We have 2 link outs in partnership with Safator, who is producing the Varsum database. When you have a variant of interest and you'd like to see some population frequency information or computational models of pathogenicity, we have link outs to go to that information in the Varsum website through our partnership with Safator. And similarly, if you'd like to perform a search through Google, there is a Google Scholar link out that would include the various ways that that variant can be described so that you can perform a search in Google Scholar. But Mastermind has the content that's required to answer the questions that you need and is a much more thoughtfully organized and clinically relevant presentation of the information for your variant curation activities. So when I click on any one of these references, either in the list form or in the graphical form here, if I click on any one of those, you'll see 2 drawers open up from the bottom. On the left is the title and abstract of the paper that you've selected, and on the right are full text sentence matches for that particular paper that are highlighting the mention of particularly relevant terms from that paper. In this case, it's defaulting to genes and variants, but if you want to see only those sentences that mention the particular gene, you can click on that window. Or in the case that I want to highlight for this use case, if you want to specifically focus on the papers and their mention of the variant of interest, you can click on and engage the variants only window here, and then rapidly toggle from article to article to see all of the different ways that the author has described the variant, and more importantly, see the sentence context where that particular variant is discussed. So if I toggle through those papers, or if I identify a reference that I'm really keen to identify and interrogate, I can click on it directly or toggle to it. If, say, this particular sentence or set of sentences is particularly interesting to me and my clinical question, and I want to take a deeper dive, and I've installed a Google Chrome extension and have access to that content, you can see how this was presented through the web browser in the Mastermind interface just now. If you have access to that content, you can click on and see and interact with that PDF using a variety of these PDF tools. And so to get back to the sentence matches, you can click on Show Matches. To restore the panels to their original height, you can click on those Expand Pane capabilities. And that's the use case for identifying the variants described in any of these papers of interest to begin your curation activities. So a version of that use case is to say, how do I identify the most seminal paper, the original paper that talks about the variant of interest, versus identifying some of the more recent papers? Well, that's where this aspect of the publication history becomes meaningful. You can see well-supported, widely cited papers that talk about your variant of interest based on how high they appear on the y-axis, or most originally published or seminal studies on the hugging here on the x-axis at the left, or more recent studies, including those that were maybe published within the last week on the right side. Similarly, if you'd like to see that in content in list form, you can click on and engage a different sort parameter, not the clinical relevance or the content relevance order, which was what is presented by default, but rather the publication date order here, either in ascending or descending order. Another way to prioritize your search, if say 70 articles is too many for you to search through, and you're curating this variant in the context of the ACMG guidelines, a version of the use case that we're discussing now is I'm a researcher or clinician interested in a variant that I've identified in a patient, and I wish to classify that variant or interpret that variant according to ACMG guidelines. So the way to do that for professional users uses this third aspect of the Mastermind interface, which is the categories, a way to enhance the specificity of your search at the keyword or category level. And because the use case that I'm talking about now involves specifically curating for ACMG interpretation, I will click on that category header for ACMG, and you'll see a full palette of different category headings. In this case, these are specific to or focused on the ACMG guidelines. For those of you who are familiar with those guidelines, some of the most meaningful content that needs to be extracted from the medical literature involves designation of strong evidence for pathogenicity in the context of functional studies. So I click on functional studies here, and you can see all of these different key terms that are associated with functional studies, broken down according to whether they're in vivo or in vitro type assays or terms associated with functional studies. And I can either click on these individually, such as I'm doing here, click on them categorically, as I've done here, or enable every one that you see on the page entirely. And if I've enabled every one of them or a handful of them, and you are able to cross categories here as well, so this becomes very combinatorial, I submit with my chosen filters, and what happens is the papers from that original list of 70 that do not talk about any of the terms that I've engaged will be filtered out, but the papers that remain will be reprioritized, as you can see here in the publication history, as well as notice here in the context of the list, they've been now reprioritized with functional studies and the associated keywords as a primary focal point. And so to identify which of these references is the most meaningful, you can click on each of those references in succession and get a sense for the content, either at the sentences that describe the variant, the sentences that describe specific key terms that you've engaged, in this case, functional studies, so things like wild type and in vitro will appear, or otherwise looking at all of them in their entirety, you can see the keywords, the variant mentions adjacent to each other and get a sense for what the authors were attempting to describe in this paper. And then again, if that piques your interest and it addresses a focused clinical question that you're interested in, you can seek out the PDF if you've installed the Google Chrome extension. If you would like to rapidly toggle through this information, one of the more meaningful ways to do that, initially at least, is to focus on the variant sentence contexts, is to focus on that in the drop-down at the right in the lower right pane, and then to very rapidly toggle through and read the information until you've identified which of those papers is most likely to contain the information that you need. In this case, this particular paper is meaningful because it has a number of mentions of the variant, and when I toggle to the keywords, it clearly is describing murine models of this particular porphyria disease that I'm interested in for my patient. And again, if that is something that you've identified as meaningful, you can click on and view the PDF if you have access to that content. Moving forward and out of the UROS gene to highlight some other features for a particular additional gene, if your question is, I'm a researcher or clinician and I can't find any literature on a particular variant of interest. How do I find information on a neighboring set of variants? So for that purpose, I'm going to the ATM gene. You can see the whole landscape of variants in the ATM gene in visual form here and in list form. If you're interested in identifying variants that are neighboring, if you want to tease apart some of these additional variants, say you have a variant in this region of the protein and you're interested in finding the variants that are neighboring, you can either do that by clicking on and engaging any one of these neighboring variants, or otherwise sorting, say, by cDNA position, as I'm doing here, and then focusing in on the region of interest, or otherwise filtering so that you're only identifying, say, loss of function variants, in this case, frameshift variants by typing in FS in the filter box, nonsense variants, as I've done here by typing in X, splice changes, splice acceptor, splice donor, or intronic changes, as I've done here. And once you've identified the variant of interest, you can click on the variant that's neighboring your variant or similar in its nature to the variant that you have in your patient, and then investigate the literature in much the same way that we had done with the previous example.